Hi everyone, today we are going to be talking about nanopore sequencer.
What you're seeing here is a protein nanopore. In nanopore sequencing, protein nanopores are embedded onto a synthetic membrane submerged in an electrophysiological solution, and a constant potential is passed through the nanopores. To sequence DNA, each DNA strand is attached with a sequencing adapter coupled with a motor protein that will drive the strand through the pore. The motor protein acts as a helicase and unwinds the DNA and causes single-stranded DNA to pass through the nanopore, slow enough to measure the characteristic disruptions in the ionic current, nucleotide by nucleotide, until the entire strand has passed through. This set of disruptions will be detected and analyzed in real time to determine the sequence of DNA bases passing through the pore. Once a strand has been read, another DNA strand will be pulled in and the sequence restarts. These processes are repeated with thousands of nanopores over a huge nanopore membrane, increasing efficiency and accuracy. Nanopore sequencing is able to sequence as long as the DNA strand is presented and therefore generate long reads.
We will be explaining about one of the simple machines for nanopore technology called MinION. For the nanopore technology procedure, the priming pot cover must be opened. A P1000 pipette is used to withdraw a few microliters of buffer by either raising the plunger gently or twisting the plunger. Then we load 800 microliters of priming mix through the priming pot using a P1000 pipette. Then we wait for 5 minutes and then open the spot on tap. Next, we load 200 microliters of priming flush through the priming pot whilst keeping the spot on pot open. As we do this, we force the buffer front in the spot on pot to rise slightly and displace any air bubbles that may be there. The library needs to be loaded onto the flow cell immediately after the second flush. Use a P250 pipette to load the library dropwise onto the spot on pot. Ensure the tip only hovers above the spot and it doesn't come in contact with the pot. After the library is loaded, close both the priming and spot on tabs to prevent evaporation and a subsequent change in osmotics during the run of the experiment. Nanopore starts to stream data as soon as the experiment begins. This data is processed by the instrument control software and can be uploaded to MetricCore, an on-demand cloud-based data analysis and bioinformatics platform. This allows the users to continue an experiment until sufficient data has been produced to determine the answer to the biological question. We analyze the result obtained from the nanopore technology from MiniNo, which is an operating software that helps in data acquisition, real-time analysis, local base calling, and data streaming. Base calling is a process of conversion of the electrical signal generated from the DNA or RNA strand that passes through the nanopore into the corresponding base sequence of the strand. By this, we can know the sequence of the DNA or RNA of interest. That's all from us. Thank you.